Hello everyone, welcome back to our channel. So if you are new to our channel, subscribe the channel and press the bell icon. So in this video, we are going to talk about the expression vectors. So what exactly are these expression vectors? Basically, these expression vectors, they are the type of the vectors that yield the protein product of the gene. So these are the variants of the plasmid vectors, and they are the modified plasmid vectors why the expression takes place. So here the expression of the gene takes place. That is why we call them as the expression vectors. Now we will see here the expression vector, the structure of the expression vector. So here we have this expression vector as it needs the origin of replication so that the replication can start. As we have seen in the previous video, different vectors, they need the origin of replication. Then they need the selectable marker. So we have seen the selectable markers can be different types like the ampicillin resistant gene, the tetracycline resistant gene, the chloramphenicol resistant gene, or the kanamycin resistant gene. And besides the selectable marker, they also need the multiple cloning site. So this multiple cloning site, that is the polylinker site. It is a site where the different restriction enzymes can cut. They can cleave that site and we can insert our gene of interest, that is the DNA insert. So this is very important in case of the expression vector. Then we need the promoter sequence. So these promoter sequences, they are important for the expression of the vector. We need the easily induced promoters so that the expression will take place. And then we need the ribosomal binding site. So this is the site where the ribosomes will bind. And we also need the transcription stop site so that the transcription will terminate at that site. And then after this transcription stop site, we need the Shine-Dalgarno sequence. That is the sequence where the initiation will take place. And we also need the Kozai consensus sequence. So these are the sequences where from the initiation will take place. So this is the overall the expression vector. And besides these things, we also need the purification tag. So the different purification tags like the histidine tag, the fluorescent protein, or the maltose binding protein, or the glutathione S transferase. So we need the glutathione S transferase. So these are the different purification tags so that we can purify the protein. So here we have this expression vector. As we know, for the expression, we need the promoter sequence so that the RNA polymerase can bind at that sequence, and then mRNA formation takes place, and then the ribosomes bind at that site, and in this way the protein is formed. The translation takes place and the protein is formed. So here we have this Shine-Dalgarno sequence where we can clearly see that the ribosome binds there at the site, and then the translation takes place. So this is the Shine-Dalgarno sequence. Applications of the expression vectors are that we can use them to purify the pure protein. So these are used in the purification of the proteins. And besides the purification, we can also study protein localization. And we can also form the recombinant enzyme or the protein production. And then we can create the dominant proteins or the negative protein. So we can create also the different types of proteins. So this is the application of the expression vector.